question that you will probably want to ask is, why compare the two genes? And in fact, there are many reasons why you'd want to do this. Let's imagine there is a new virus that comes out. You'd want to know how to counteract this virus, but you won't want to start from scratch. One method of actually trying to find a medication or some sort of treatment against this virus is to take this new virus and compare it to all of the genes of known viruses from a gene database. You sequence it. You align the sequences. Once you compare them, you find that you may, if you're lucky, have another virus which is very similar to this virus. Then you can take the treatment of the virus that you know and use that as a starting block for finding new treatment. There are other reasons that you want to use sequencing. Sequencing can be used in finding out our history. Have we actually evolved from apes or not? How close is our genetic makeup to other types of animals? With genetic information, you can actually find out which place in the world your HIV infection actually originated from. You can know the different which subtype of HIV you may have. You will also want to compare genes because you want to know whether some sort of virus or plant species is actually new. Is it a new discovery? Is this a new type of animal? Or is it a mutation? Right now, I think what we will do is have a demonstration where I show you one example of sequencing, and that has to do with HIV subtyping. We will go to this BioAfrica website, which has a tool that actually sequences genes with a database of HIV viruses of different subtypes, and then tries to find out what subtype this unknown sequence is. Is it a single subtype? Or is it a composite subtypes? So this is the BioAfrica website. I went beforehand like a good lecturer and copied a subtype, a gene, simply put it in there and click run. So what this is doing is it's taking this entire gene sequence here and it's now aligning it to each and every gene sequence that it has in its database. Each gene sequence in its database has a known subtype. It's using maybe BLAST, maybe a combination of other sequencing algorithms to find matches, to find the sequence or sequences that this large sequence actually matches to. And of course it takes a long time because these are complex mathematical processes, and here this is done. So let's look at the report. And you find that it shows you that in this gene, which was actually a pole gene, it found subtype G and subtype A sequences. So this green here is the subtype G sequence or subtype of HIV. This here is the subtype A HIV, and this is again the subtype G HIV. So in this sequence which is obtained from an HIV patient, part of the sequence is subtype G, then a part of it is subtype A, and the rest of it, or another part of it, is subtype G. So now there has been studies that have shown that treatment for HIV patients do differ depending on what subtype the patient is actually affected with. So if you now can use a sequencing tool like this and determine, okay, my patient has a combination of subtype G and subtype A HIV, what treatment can I use to best help the patient reduce his viral load? The second example that I want to show you is the Stanford HIV drug resistant database. Now, what this website does, well, what this section of this website does, is that it allows you to tell it what mutations have occurred in a genome, and those mutations are associated with specific resistance for HIV. So let's start a little bit from the beginning. When HIV infects a patient, it inserts its genetic makeup into the patient's genome. Now, assuming that you have the wild type HIV, all drug cocktails should work. But over time, because of selective pressure, or you may have been initially infected with a particular type of genetic makeup, you may become resistant to certain types of drugs because your gene, the gene that has been inserted into your genome, into your makeup, has changed. Certain mutations are associated with certain drug resistance. So what the researchers at this Stanford HIV database have actually done was they found out what mutations actually cause resistance to particular drugs, and this is one form of bioinformatics. So if you look at this example here, if I have a mutation at a different points in my gene expression, let's put 2 more, what happens? Analyze. So what will normally happen is if you have a gene sequence, that gene sequence is then analyzed or sequenced against the wild type HIV virus, and you find out at what positions you have mutations. You then use a bioinformatics tool like this, enter wherever the mutations occurred, and it will give you a resistant report. So it says here, you have intermediate resistance to the drug ABC. You have low level resistance to DDI. You are susceptible, that means the drug FTC will actually reduce the viral load in your body. You have again intermediate resistance to EFE. So you can now start understanding why a bioinformatics tool like this is important. If you can gain access to a patient's genome, so you get a blood sample, you sequence it, you then compare or sequence or align the sequence against the wild type HIV virus. You find out which positions there are mutations in, you find out which drugs the patient is resistant to, and you don't add those drugs to the patient's cocktail. And if this is done, you actually produce a much better standard of care, and the person's viral load decreases much faster. So that was the HIV DB database and the sequencing tool. Both tools sequence the genome, and then the HIV database goes a step further and analyzes the alignment. As we mentioned earlier, another important aspect, another important use of alignment is actually finding similarities. Similarities between different genes for many reasons. Then we spoke about different viruses, but a very interesting one to me is actually finding out where we've come from. How many of us actually believe in the theory of evolution? Have we in fact evolved somehow from apes? And can bioinformatics maybe help us shed some light in this. Right now, I'm going to show you two other websites. The first website that I'm going to show you is the NCBI website. This website is a very important resource for people that are studying bioinformatics. It's actually a gene bank. It's actually a database that is full of sequenced genomes of almost every single type of organic matter that you can think of. So all you simply do is choose the type of structure you want, whether you want nucleotides, whether you want whole genomes, whether you want proteins. So let us choose proteins, and then you search for what you want. Let's say we want to try and search for the chimp genome, right? So let's start off with the chimp genome. I have the name of the chimp genome here. Let's choose that. Go here, click there. So this is actually choosing a certain genome that's actually Homo sapiens. So this is a human genome. Go to search. So it found. Let's choose one of them. Now, this particular tool, which we are going to use to try and find an alignment between the genes, uses the FASTA format. The FASTA format is simply a format in which one exchanges genome information. It's simply a format that is standard way of describing the genome or a certain part of the gene. So if I copy that, I go to this tool here, Clustal W2, which is the tool that is going to find out how close the two sequences are. Let me go back. Here, I want to find a new gene, a new protein. Let's see whether I actually have it so we can save time. Homo sapien. Well, I actually do. This is the genome of the chimp, which we are told we evolved from. Oops, why did I press browse? Thing to do. Cancel. Go down and submit. Okay. This will in fact take some time to do because the actual process of alignment is very complex. Okay, so we see here we have the two sequences aligned to each other. Wherever there are stars, it means that the sequence matched. Wherever there is the semicolon, it means that there was a difference. Let's view this in result summary. Okay, so we look here, we look at the score is 94%. So what does that mean? That simply means that the two genes are 94% similar to each other. And if you look at it, most of it is stars, which shows that they match in those positions. There are very few semicolons and other changes in the genome. So that actually proves to us that this part of our genome is 94% similar to that of the chimp. And this, of course, was done with the entire chimp genome to actually try and prove to us that we, in fact, evolved from chimps. Now, the next question you might ask is, you're looking at this picture, why do I have a picture of a pig? Are we evolving into pigs? The reason why I did this is because I actually want to show you how close are we to pigs. So again, if I can, let's just copy this, paste this here, and let's put the pig. Can you guess how close we are to pigs? 60%? 50%? Think about how our skin looks, whether pigs have hair or not. Let's choose that one. We want the faster format. There you go. Copy that. Let's go to cluster. Go back one. We want to take out the chimp. And let's put in the pig. Let's submit. It's verifying. It's comparing the genes. It's going. It's thinking. It's aligning the sequences. And here we go. A bit more differences than the human and the chimp. Let's go to result summary. 76%. So for this part of the DNA that we've analyzed, a pig and a human being is 76% similar. So the next time someone tells you your room looks like a pigsty, you can very, very confidently tell them, yes, that's true, and that's because I'm 76% pig.